It's a modified line inside. Okay, so again, make sure you. Why the numbers? The last is.
Yes. The plates are going to be there. I will explain it in the time. I just want to fix one projector. For now, it'll be. Folks, there are some materials that you guys can have, especially when it comes to actually, you have most of the. I would say start off with sterilizing the desktop. Yeah.
Oh, my brother needs it. Okay.
Yeah, that's very nice. I think we're good for timing. Begin shortly. I'll wait another 2 minutes for those that just came in. And for those that don't know, hopefully you've been keeping an eye on my messages. I said this about last week, if not a week ago, a week before, about presentations. So this week you guys have presentations. I hope many of you are aware. Some other TAs may have sent it more recently, but I try to send these types of tips in advance so you guys are able to, once again, prepare and excel accordingly. And of course, if you are wondering, what am I going to essentially talk about for about 5 to 10 minutes? It's pretty much the analysis. Types of chemical germicides, your results at time 0 to time 120, and then having another pair of your members of your group talk about the antimicrobial susceptibility test. You guys don't worry too much. I want to see how well you guys know your material. That's what's more important. You guys can use your notes. You guys could use even a manual or your notebook accordingly. And I don't mind if you have to pause and coordinate essentially your presentation with one another. But that's to come in due time. In the meantime, we have lab material. Right, exercise 1, lethal effect of UV. We're going to be looking actually the UV. I have to turn it on for you guys, so I'll do that shortly. Exercise 2 will be on radiation. And so essentially, you have the materials with us ready. So, you have different plates around your desktop, different bacteria as well. To start off, let's go straight to exercise 1. Before I do any questions, folks, based on what I've talked about thus far, I also want to highlight some of you, and this is a common trend, but you guys have to be vigilant on this regard. Make sure that your figures are detailed with captions, because this is something that will also reflect your proficiency for an upcoming FLR. So you have a second one on the catalase test and antiseptic/germicide results, which hopefully, inshallah, Dr. Brent will share more details soon. Let's see. Go forward. It's moving around. Let me just go here. So slide 2, lethal effect of UV. You guys have 5 different bacterium on your bench tops with red trees. And if you have a look, 4 of them have specific details of not just the bacteria, but of the age. So bear that in mind because you're using only 4 out of the 5 that have specific references. M. roseus, Microphocus roseus, the one that you're not going to use for the first assay. However, I want you to be able to distinguish the different properties of pretty much what you're about to do today. So we just look briefly into, I think there was another detail. Oh yeah. Let's go up. So each of you guys should have a set of slides. So sorry, plates. Your plates for today on your bench tops are for exercise 2 and for the post lab of last week's antimicrobial susceptibility test. For exercise 1, the plates are in the middle bench. And so this is where I just want you guys to all pay attention because I don't want to make mistakes. Imagine you have you separated, right, like Microphocus roseus, because you're not going to use it as of yet for now. But you notice they have 4 different other bacteria or different cultures with different ages or time points for each dish. The idea is, like what we've done before, you're going to be taking half of the same, this half in volume, to pour into each of those 4 tubes for exercise 1. And then the goal is you're going to be essentially plating them for each. Oh yeah, so I'll talk about that. But the point is that for the 4 different bacteria, one's going to go with each plate. They're going to be spread plating like we've done in the past, inverting or trimming 1/3 each time. And then the goal is, following a separate technique, you want to make sure that you obviously the plates are able to follow up with your need. And each of you guys are going to have essentially a set of plates, and you're going to be essentially accounting for the different time points that they're going to be placed in UV. So you're going to have like, and let me know if I'm too quick. You guys are a bit confused. Can I hide this? I'll show you what I mean in just a moment. So if we go, for those that want to also keep up, page 106 of the lab manual, you guys are in different groups, as you can imagine, 1, 2, 3, all the way to 6. But I want you guys to be able to essentially do the exposure times for your for your specific time point of interest. So you're going to be essentially, once again, placing in half of the volume of saline into each of those 4 tubes, and then following a septic technique, pouring them, taking a sterile swab, and essentially, let's talk about it because I don't want to waste time. The idea is that you want to be able to take that swab, spread 1/3 each time on each of those plates so you guys can do it in pairs. And yes, inshallah, then go straight ahead to UV exposure. Any questions regarding that? I'll separate you guys into groups in due time or in terms of the time points, but you can see them here. Group 1 is going to be no UV, 5 minutes at UV. Group 2 for 10 minutes, Group 3 for 15, Group 4 for 20, Group 5 for 25, and Group 6 for 30 minutes with UV exposure. Hannah, any questions?
Yeah.
Yes. Yes. So actually, this may help because you may not see it in the slides itself. So this may be a bit explanatory for those that don't have it written down. So just have a look at this slide. And I would say get started as soon as you can. In terms of preparing? Yes. Sorry? Oh, the foil. Foil? Is that your question? So great question. Folks, we have foil here. We have aluminum foil, but I would recommend using these ones because there's so many plates that we're dealing with today. They might be used for ones that are only serving as aluminum caps. For exercise 2, we have the star ones here. I think we'll also bear in mind because whenever you do these, please have these goggles or safety glasses, safety goggles. You can put them right. And the other thing I wanted to highlight is that you guys want to do this. Yeah, so label accordingly your plates with the name of your group, the practical, right? Because essentially some of these, at least for exercise 2, you're gonna have to place them down at that end. And so you need to be able to identify them, right? So just as a kind of reminder, your practical, wait, exercise 1, 2. The time points. So conditions, right? So you already know the conditions. It can be the time point, like from a plaque assay, solution factor, etc. And the date is March 15, 2025. So yeah, with that, just get it straight ahead. Just get started. The gas is on. You guys can flame accordingly. Grab 3 vials of saline. Grab 3 vials. Each group can grab 3. And you'll be able to make use of the last one for Micrococcus luteus. Make sure as well, folks, that you don't bear in mind that you don't go over this gland when you're adding in the saline. It should be around like this level. I know it may be a bit out of scale, but try not to overdo it, so that way you get a concentrated mixture bacteria. Sorry. No. Don't use so folks for the plate. Yes. So just to clarify, use the plates that I showed you here for exercise 1, exercise 2, and from last week, the ones you already have, just go there. I'll get to you, Scott, in a moment. Do you have a question? Okay, okay. I would always see it. So it's a yes. So even if it's expressed, maybe like this is figure 1, for example, or like in this case, figure 6-1, 6, talking about that this was your observations of catalase test using H2 hydrogen peroxide and specifying the distributory proline, and any additional conditions that you have. Because that way even just to gauge the level of quality of your book for the grade. Have any questions by me? No. If anyone has questions, let me know. I'm gonna put a timer for you guys. Try to be done this step in the next 10 minutes. So yes, and that's another point. Make sure to label your bacterium as well. Oh, it includes results. So like figure captions, like your results, let's say beta-lactamase test, antimicrobial susceptibility test, catalase test. And then for each of the tests that you have observations that are visual, then you include the figure captions.
Okay, well, if I forgot to do that because I did why results.
If you can send it to me as soon as possible, that's okay.
Okay, thank you so much.
But for the future, try to do it within the same lab, so that way, or at least before the lab, so that way I'll be able to check it off as soon as possible. No. There was another question. Sorry, someone came here? Okay.
I thought it's something about it.
You guys need any additional materials to have sterile swabs here that will be useful to essentially swab accordingly your bacterium. Try to make sure that they're mixed readily. And I actually am going to go through a few other things as the time goes down. I didn't talk about why you were using those 4 different bacterium, but I want you to think of your unknown assignment once again. What's the difference between E. coli and Staphylococcus epidermidis? Anyone know? Which one's positive? Oh. Okay, B subtilis is positive. Yes, Sahar. Is E. coli positive? Negative. So gram negative for E. coli. Remember this, folks. I don't want you guys to get confused. Gram negative. The only one that was gram negative from our unknown assignment was E. coli. Pretty much on the right hand side. Gram negative. All the other ones, B. subtilis, Staphylococcus epidermidis, Streptococcus, pneumoniae, etc. Those are gram-positive bacterium. And so the idea is that they have differences in composition, peptidoglycan layer thickness. And so that hopefully will give you an indication, inshallah, of the effect that it has on UV, right? And then now the second question I have for you guys, the difference between B. subtilis at 24 hours and 7 days. Great. Now let me ask again, in terms of difference, 24 hours versus 7 days. Where would the spores be present at which time point? Yeah, between like, so which one, like, if you were to do the ended spores chain once again, and you had two different cultures at 24 hours and 7 days for bacillus, would you expect it to be capsule, sorry, endospores inside bacillus at 24 hours or at 24 hours? You guys got it right, actually. So great job. 7 days, essentially, it would be outside of the bacterium. So they would have been exocytosed. And so that, therefore, the function of endospores, as hopefully many of you know, relates not only to, actually, let me ask one more time. What's the functions of endospores? Anyone know the function of endospores? Seeds, okay, carrots as well. What were you planning to say? Sorry? Not necessarily. This one's actually different. So, and place close attention to this because the capsule stain is different from the endospore stain in function. Capsules are the ones that have the waxy layer, like layer, that's at the outer coat. That's beneficial for biofilms, which we may be able to talk about near the end of the term, but great response nonetheless. So happiness. You have something else to say? Oh, okay. You didn't or you do? Okay. So to clarify as well, endospores beneficial for UV resistance, beneficial for resistance against hot temperatures and pH or salinity differences. So think about like, different bacterium that live, let's say, in aquatic environments that are thermal, like very hot indeed, like the hot pools or essentially even in the Arctic, you have different extreme temperatures and conditions. And even in like hypoxic or hypersaline environments. So those are things that you want to just bear in mind. Yes. And another point is when you're at the stage, inshallah, of going forward to the UV, bear in mind that you have soaked or have at least placed and sprayed paper towels with ethanol. And thereafter, you're opening up your plates with the lid exposed at the bottom. So just bear that in mind. If you guys are having problems, let me know. I see that one of the group 2, is already using the UV or preparing accordingly. When you guys are ready, let me know. Try to be done in the next 2 to 3 minutes. Folks, in case you're just wondering, I said this before, but I just want to repeat, with your swab, you're going to do this 3 times. So inverse or turn 3 times, right? Just like what you've done for spread plating, but you just want to make sure that you're going to essentially cover each plate with your specific bacterium of interest. When your group is done, let me know. If your group, by the way, if your group is already started, have a timer. So in the meantime,
Yes.
How are you guys doing thus far? Anyone, I know who is good. You guys, you can, yes. The issue is though, I'm gonna give you instead the antimicrobial susceptibility test, please. Reason that I wouldn't recommend UV as of yet is because, as you can see, we're gonna be using the same time for the other time points. So the other groups will also have to do it in 2 times. But for those that are waiting in the meantime, you can press up for the germicides. I'll try to see it for you. And then the antimicrobial susceptibility test. Give me just a moment. If you guys are difficulty out of your plate, you guys are having difficulty arranging your plate. That was quite long. How's that going to do? Thanks for your time. Almost, but it's completed. Yeah.
Yeah.
Folks, if you are not as of yet done, I kindly ask and request that other members of your group also help accordingly. If you guys need help, I'll try to help you guys. But if any other groups are like having people that are vacant, that's a problem. Make sure everyone's being active and at least doing one exercise, if not more. Yes. No problem. Well, 2 important points. We have space for 8 plates for each UV lamp. So if your group is waiting, you can actually set it off, turn it back on accordingly. That way other groups can accordingly use it with your kitchen. So other extra plates. Other so yeah. Yes. So in the meantime, make sure everyone is placing in their place as efficiently as possible. So, for example, I think space for another group right now. We can start with have it on the corner, and then we can do it. That way we can use your control, by the way, does not have YouTube. Yes, the chair is. I noticed that some of you may have not taken note of something very important. So if you look at the manual, okay, this side has it, but this side, I need to make sure you guys are aware. You need paper towels, soap couple, ethanol, underneath, and so I'm actually.
Future if they want to switch the lab.
And maybe perhaps something that we can. No, it makes sense. I'll be right back.
And Switzerland is the most.
Sorry, guys. In this not ours, only to actually group session better. And that's what I was thinking about. So So for our lecture, well, you know,
No, but we have. Yeah, it's in the presentation. Yeah, today. So I can say. Yeah. Restaurant.
Does anyone have excess ethanol with them? Some groups are in need of extra. Anyone have extra ethanol? So you guys wait, apparently this so keep it improved and you can how many more groups are waiting to turn to place their plates under the UV? Okay, okay. Any other groups? I want to ask what's the timeline for the groups here? How many more minutes? So in the meantime, for 6 months, you can use lactate. Well, some of you may be wondering, what's the presentation about? So maybe, I guess, now would be a good time to talk about it. So I'm gonna just show you to make it easier. I have, so for your chemical germicides, have a look at what was posted on Perkis. You'll also see it on the slide when you're doing the presentation. So I'll show you what you're doing. So this is your results from practical 8, right? And essentially, you had all the different time points. And essentially, my request is that you essentially compare at time 1, which is technically 0 minutes. Just compare that value with your results here at 9, at the 9th time point. In fact, actually, because of this is for the chemical germicides, yes. In fact, you actually could just look at the results here for context, because you have the positive, negative control, the lowest, middle, and highest concentration of your germicide. So just talk a bit about what your germicide is an antiseptic, disinfectant. Let me just write it down actually for you guys. But you guys pretty much have the results that you need. And then for your antimicrobial susceptibility test, you guys need to, once again, think of it like a mini FLR. You have an intro, results, discussion. You're skipping the methods, but briefly talk about what you've done and what results you've uncovered, and as well as what those results imply. So like your conclusions and discussing any limitations from those results. Especially the germicides. Germicides and catalase tests, unless Dr. Brent changes it.
I was looking at the name of it, it said UV damage.
That's what so it may be the case, but I saw an email that said otherwise. So maybe just to be safe, go.
I think last year it was germicides.
Last year was, if I'm not mistaken, but nonetheless, what you see from the manual. But I do know that the catalase test is included. So that's something that you may want to. Sorry? No, I'll talk about that when there's due time. Don't worry about that too much. I know many of you are probably wondering about the generation time requirement due next week at the beginning. So think about that in due time. In the meantime, for your presentation, let's see. Start with okay. Let me just see timing. Yes. No, all of you should be talking. So essentially, but it's relatively simple. So let's say the two of you focus on the antimicrobial susceptibility test, and the other two of you focus on the germicides. You guys can split up. One of you talk about the intro, the other speaks about the results, and the last person or the person that spoke about the intro talks about the conclusions. Because the results takes a bit of time, intro and discussion, conclusion is relatively simple. Let me just give you some thought. In the meantime, just pay attention with what I've said thus far. So antimicrobial, this is like new, but in my work, it's like microbe. So let's just say microbial tests. So talk about the same applies to your germicide. Talk about what your experiment is, what your research question is, you know? Just trying to see. Okay. In the meantime, you guys can continue with exercise 2. I'll talk about it in about 5-ish minutes. But if you guys are essentially ready for exercise 2, start labeling your plates. For the germicide, just focus on your group's results. You don't have to do all tables, just focus on the column. So remember group 1, your group 1, 2, 3, 4, 5, 6. If the groups on that side can't see, let me know. Someone's Bunsen burner was just a bit too high. So think about it like this. This records your absorbance values. How turbid your culture is.
Not bad. The higher concentration.
If there is more turbidity, I still smell it. How much Bunsen burner?
Ours is on, ours, we're doing plating, so line it on.
How high is it?
On how you want
Anyone else? But it would be for both sides. Both sides.
So you are correct. Hydrogen peroxide. So first one was like fix. So why did it decrease from the go the opposite?
Where are you looking at?
The concentration of it. But everybody else needs anything. Wait, that's how
So what I would recommend is see if there was any errors. Does this match what you just affected, you know? And what should have happened? Well, did you say the highest, the whole concentration on the soil?
I thought it was the lowest one.
Yes.
But I guess the highest one.
Yeah. It's really the drive of limitation last time.
This is an India code.
Actually, it could be. Also,
Especially
side and then
One that we're doing was one part two process.
this results signify that it is sense. So now, as oh, it's already on.
Yeah, for exercise.
Okay, perfect. Sorry. You can take it off. So now let me just go straight ahead to exercise 2, talking a bit more. Let's see. Actually, yes, okay. Exercise 2. I know maybe some of you are already ahead, which is amazing. Keep it up. But just to reiterate this, in case you're concerned and have some questions, you had 7 plates on your bench, plus 2 for another exercise. The 7 plates, as you can see on the slide, are being separated for each condition. I want you to all pay attention to this because these 7 plates. So bear in mind, if another group is paying attention to antimicrobial susceptibility test or on exercise 1, then the remaining number of you can focus on this exercise. You have 7 different plates. I'm just going to make sure everyone can see, so I'll bend a bit. You have, unlike what you've done for exercise 1, looking at multiple time points that differ about 15 minutes in turn. In this regard, every 5 minutes. This one you're only doing in a finite range, each group from 0 to 3 minutes. And there's specific controls and specific conditions that are needed to be met. So if you look at, let's say, conditions for plates 1 and 2, both are going to be under UV for 30 seconds. So make sure you have a timer on. But the difference is pretty much what we've done and following the adjusted or the corrected procedure today. You can have your lids essentially following the procedure as you can see here. The difference is that for conditions 1 and 2, both are 30 seconds with the lid off at UV with UV. But the difference is for incubation. Actually, let me clarify this before I move this. You guys are going to use Micrococcus roseus. I talked about that earlier. You're going to follow the same technique I spoke about, but you're going to use these star mats, the ones that are in the middle bench. And so somewhere you'll be able to essentially not have to use the aluminum foil, just use the paper masks. And then accordingly, essentially masks where it's shown, i.e., place 1 to 5, all use those masks, and place 6 and 7 are pretty much your controls, with or without light incubation. When it's light incubation, it means that you're going to just place your plates, the ones that have light shown 1, 3, 5, and 6, they're going to be down there at the very end. So have a look, and essentially, I just want to make sure you guys, because there's so many plates there, make sure you label them and arrange them in an organized manner, so that way all 4 of yours are stacked in one line or one column. But the ones that are shown here, let's say for the dark incubations, you can actually wrap them as a group, like the 3 of them, in aluminum foil. So just wrap them all up. Make sure the aluminum foil at the very end is labeled as well. Practical, exercise, group number, and date. Or even just without the dates, because kind of explanatory. Place 3 to 5 are going to be for 3 minutes that you need. So bear that in mind. I would recommend try to do the plates with specific incubations, i.e. 30 seconds or 3 minutes separately, so that way other groups can take their turns accordingly as well. But just keep them as you've done for exercise 1. Leave the agar at the bottom, or like place or position your plates such that the agar is at the bottom as you normally spread plate, and then you should be able to take care of this. So any questions, folks? Exercise 2. I know some of you, can I have a show of hands? How many of you are just waiting for exercise 1 to be done? 1, 2, almost done? They already placed theirs in. In the meantime, though, all other groups are done exercise 1, just to confirm. Perfect. Done exercise 1? Yeah. Okay, so now you guys can get started for exercise 2. And I know for the group that is waiting, let's see, you can get started with the antimicrobial susceptibility test, the 2 groups in the meantime, so that way you guys can prep up for your presentations. And in the meantime, you guys can also make use of the time that you have. I once again just want to reiterate this. A lot of these tasks are meant to be done as groups. So make sure that everyone in your group is active. Very important indeed. And if you guys have trouble or you're seeing that dynamics aren't working out as well, just let me know. And I'll be more than happy to help. But I just want to make sure that even if you, let's say, are preoccupied and notice that time is running low, have members of your group split up and coordinate the remaining exercises. Thanks, everyone. And I'll give you 15 minutes before I give another or ask for another update. Any questions, by the way? Yes, morning.
What's your occupation?
Let me talk about the antimicrobial susceptibility test. Oh,
So which one's called?
Epidermidis would have been looking bigger, so it would have had essentially higher. Like it's a bit relative.
I'm gonna say the fact is.
You can tell me about what you would have expected and why you want to use.
I'm like 9%.
I had a feeling it wasn't necessarily the order, it was maybe a step. It could have been the color, being potentially user-friendly or close-fitting. Normally you want to take a particular, so that's going to be one of the common variables, such as there is a yeah. But it's okay. It's okay. I'm not penalizing. I'm not penalizing. Even if it's in the SLR, hypothetically, like don't worry.
It's all good.
And like the experience. But folks, let me just go straight now to the antimicrobial susceptibility test. Some of you guys are straight ahead, which is amazing. Congrats, folks. Now, I guess I should talk about this slide as well. For similar to Micrococcus roseus, so following UV, you're likely going to see next week different colored colonies. Some that are red, majority of which are your normal colonies. for Micrococcus roseus. As we see, the species Latin name shows roseus, indication that it's reddish in color. But the white colonies indicate actually mutagenic colonies, or essentially those that have undergone stable mutation. And so even though this is under like, I don't know, like under a minute for many of them, you can kind of appreciate how deleterious UV can be for specific colonial growth, but also the increase in resistant or mutative colonies. So that's something you want to bear in mind. And as we move further, that's for next week. So like post-lab material. But for essentially, there's actually two things that you guys need to do for the antimicrobial susceptibility test. I'll talk about them after 2 minutes, so that way you guys can complete any final materials. How is it going? Okay. Yes. I want to see if we can get you guys now into doing exercise. Is that okay? Yeah. Folks, how many of you are done exercise 2? Okay. Okay. Groups 1 and 3, are you guys almost done? Okay. My advice, use that lab because I don't see any plates there. So you guys can go straight in. I'll be talking about the additional analysis later on. I just want you guys to focus on completing exercises 1 and 2 and additional tests of analysis for today. Try to be done by 10:30, folks, so that way you can pay attention with the concerns or final points that I would share for you guys for your presentation.
So it's my fault.
They're going to be in the same presentation. But 2, 3 of you need to, right? Not you. No? Okay, so have you can perhaps do one exercise like antimicrobial or from your group.
Oh, so, okay, for example.
You have to present both. So you in your groups, you guys separate tasks. If you wanna, I know it's just 3 of you, but you guys can work out to see who's going to talk about one exercise and which of the other 2 of you will focus on the other one, and then be able to describe the points that I have here actually. Intro, the results, discussion, /
And germicide was. Germicide was the catalyst. All right. It's about.
Your column 3. Remember, like you can also look on if you have your phone. I just realized I don't know if you guys are checking columns. I mean, how about this? I like in due time. Once you guys are done these major exercises for today, I can show it back again. And even when you present, it's going to be left on. So don't worry too much. I may try to separate the slides so you guys can see this as well. If you wanted to talk about, like, if your results are similar. But don't worry. It's all. Yeah.
Yeah.
Oil.
So yes, the foil, I'll tell you guys when you get it. So if you're done, your plates are gonna actually fill these plates, and they all have to be. Aluminum foil is here. Just bear in mind, everything is self-cut because they really get sharp edge. If you need help, I'll help you accordingly. But if you move here, this is where you can actually place your plate first, like the lights are here, and you can move that your dark incubated plates on the edges. I recommend shading your garden like that way you can register where they feel that they're all together for you. We don't show that slides in case others are also on that page. Okay, time is up as well. Yeah, so pretty much plate 5 is the one main slide that you're actually keeping the lid on. It serves as a control. So that is essentially if you actually see UV effects still with a essentially barrier, plastic barrier on top of your plates. That's something that you just want to think about. The purpose of each of the different plates. Having light on and light off highlights to you the UV repair mechanisms and light repair mechanisms versus dark repair mechanisms. And then essentially the duration, how it influences colonial growth and resistance, and essentially applying the mask and to be able to control for other sections that have not been impacted accordingly. And then the lid to serve as a physical barrier between the UV and your bacteria. Now, just as an update, how are we doing so far, folks? Almost done? So let me go straight ahead now. I'm going to talk about your antimicrobial susceptibility tests in a bit more detail. I'll move down. So when you're going to be, looking at your plates, right? Some of them you may notice have within your 5 sections for each bacterium. Some of them have what's called zones of incubation. Folks, if you can pay attention, that'd be really appreciated. As this may relate to your presentation as well. So you have 5 sections, right? Ampicillin, penicillin, chlorphenol, tetracycline, and streptomycin, tetracycline, and the penicillin family. So we have this for E. coli and Staphylococcus epidermidis. You're gonna do 2 things. You guys are gonna look at the plate themselves, observe where zones of inhibition exist, right? You may also notice the size. So take these notes in mind for your post lab. Observe the range, how wide those zones of inhibition are, and also be able to compare the features of that zone. So what I mean by that is that some of them are not only large and small, some are smooth, some are jagged. If they're smooth, actually I want you guys to think about this critically, because if you look at page 91 in the manual, you'll see how that relates to next week's lab. Because the goal is, after you've observed these results, you're going to try to make actually do subculturing in those other 2 plates, the Muller-Hinton ID and H labeled plates. And the goal is essentially doing what we did earlier today. And in the remaining last tube of saline, you're going to be using your cotton swab, and then thereafter, you may have 6 sections, because it will include your positive control and the remaining other treatments that show zone of inhibition. If you have less than 6, that's okay, but essentially we're going with 6 here. You add in your saline, and then place in, for the positive control, your colonies for smearing have to be outside of the zone of inhibition. Whereas for your other treatments, those that have zones of inhibition, you're taking in your saline swab into within the zone of inhibition. So that thereafter, once you're done that, after you've essentially smeared, like you've done before, your bacteria, you're going to be essentially spread plating it. In this case, if it's outside of the zone of inhibition in your positive control, which would be done here, right? And if it was for the other ones, then you would take it from inside and accordingly smear.
This determines bacteriostatic or bacteriocyte.
That's correct. And so that's another thing that you guys will be able to hopefully see next week to more discriminately determine if it's cidal, i.e. if the effects of your antibiotic are bactericidal, i.e. it kills your bacterium, or bacteriostatic, meaning that it essentially reduces the ability of your bacterium to replicate. So it doesn't necessarily kill them, but it makes them stagnant. So think about like compared to the generation time, right? Because you have 4 different stages. You have lag, sorry, this is initial stage, log, yeah, actually lag, log, stationary, and then death. So the idea is that you're going to see for bactericidal effects, more of a cidal effect referring to like genocide, for example. Like these cidal effects refer to killing, whereas static refers to a more like a static effect in that it doesn't kill the bacterium, but it inhibits their ability to replicate, and so it remains stagnant accordingly. You can also determine this actually by the shape of your zones of inhibition. I touched upon this before. If they're smooth, that indicates that they're cidal. If it's jagged, that indicates that it is static. Because it's smooth, if you think about it, it would depict a clear zone in which bacterium are no longer present, likely. And so that would thereafter depict for you that these bacterium will likely essentially, following your analysis, be essentially that for next week. Whereas the ones that are essentially jagged results that appear essentially like
What if it's like a gradient?
triangles.
What if it's like a gradient, like it's really clear
Usually it'll be like oh, this could be a great question.
Rest will be silent. It's just like.
I would say so. I would say so. But you'll notice some of them are clearly, because remember, you would have done smearing consistently across your plate. So you'll notice some of them appear jagged, and with it being jagged, there is the ability of bacterium to coexist in that region, but they wouldn't no longer be able to reproduce or like replicate. So thereafter, that would indicate to you an idea of static effect of that antibiotic on a particular bacterial species. So if you have a look, the ones that poke out, yes, Staphylococcus epidermidis, you have jagged here, you have jagged here, you have smooth, and I believe that's
Jagged?
it's hard to tell that one. Jagged. And when it's jagged, it's like static. If it's smooth, it's sidal.
Vital is in killed.
Yeah.
Okay.
But technically, this is more like a visual check in advance for next week. Next week, after you're done, inshallah, the incubations, you will see the results accordingly. And thereafter, you'll be able to compare. Indeed, if and by the way, take a photo of your plates before you throw them, so that way you can actually compare the results accordingly. That should suffice. No problem. Yes. Let me just clarify. No. Wait. Yes, at 37 degrees. There was one that said room temperature. I'm just trying to remember. Number 2. Yes. Yes, number 2. Thanks. So in the incubator, so actually folks that are waiting to place their plates into the incubator, you can. I do want it to actually go up to 37. So if you want to wait, just invert them, and then we can talk about your presentation soon. Let me just find out. Okay, I'll talk about this now. Yes, record the observations, zone of inhibition. If you find them present, you can make a table. I prefer you guys drawing them out as well, so that way you can practice, visualizing the effects of the bacterium. Definitely take a photo. But folks, try to do this final exercise in the next 10 minutes. I'll give you 15 minutes in case you're behind. So that way we could go straight ahead to the generation time assignment and then your presentations.
And I put it in the incubator or individually?
This is for exercise 1 or 2. Together in the incubator. But for exercise 2, have them separated as rows. So how are we doing, folks? Exercise 2 in exercise 2. That's okay. Exercise 2, group 1, are we okay? You guys are done? Group 5? So if your groups are done, and you're also done the antimicrobial susceptibility test, you could get started with preparing for your presentation. And so remember what I've talked about, intro results discussion for both exercises. You could split up. Try to be done your presentation about 5 minutes. So I just really want to see that you guys are able to engage, understand the material. And inshallah, I'll ask you questions. So don't worry, I'm not I want to see you guys participate and be active in understanding or engaging in the material. So continue as you are. And if you have questions, let me know. Group 1, are we done exercises 1 and 2? Are you doing the antimicrobial susceptibility test? Okay. Are you guys working in groups? Are you splitting up? Yeah, okay, perfect. Yes. That's okay. That's okay. You guys are done the antimicrobial susceptibility test, right? Oh, you haven't done that? I see. Okay, okay. If you can, Scott, if you can leave the timer with Karis, would you mind to work on the antimicrobial susceptibility test? Is that okay? Perfect. I want to see if I could help you guys. If you guys need help, group 6, if you guys need help, let me know. At any time. I realize because yeah. If you need help or if you want me to ask another student that's less occupied to help you guys, I don't mind. No problem. Forceps should be on your benches, like tweezers. Look about, each group should have them. If not, you can also look at the back where the pipette tips are, where the white tray is. I would not recommend taking out the discs until you're done the antimicrobial susceptibility test. You don't even have to take them out, actually. Reason I say this is because you are essentially trying to take out your bacteria, either at the zone of inhibition or for your positive control outside of it. So you should have space, generally speaking, to take them out. If for whatever reason it's very difficult, and essentially, even if you don't have a zone of inhibition, you're not even taking that section. But if you forever need, whatever reason need it, let me know. Because we don't really need it. Yeah. Oh, that's so remember that your bacteria, like you open them, this is why I was concerned about the UV, but remember they're live, you may even smell the odor from them, they're metabolizing, right? So just bear that in mind that you don't, remember aseptic technique always, and to attend to your Bunsen burner when in use. Yeah,
Concentration on Germicide,
Not handling the bacteria. That is.
So, folks, before you begin your presentation, bear in mind you've done all three exercises. That includes the first two that you see on previously on the screen, the one that we dealt with, lethal effect of UV and UV repair mechanism, and also the third one being the antimicrobial susceptibility test subculture in the other two plates. Try to do that all in the next 7 minutes if you're not done. If you are done, we could go forward with talking about your generation time assignment for those that have final questions regarding it. I could give you guys the time of asking these questions now if you want. If anyone has questions. Subcultures, so I said subculturing, and it's essentially you passing bacterium from one plate to another, or one flask to another. And that's pretty much what you guys are doing right now. Similar to what we did in the unknown assignment, where you took your unknown bacteria and put them on NA slants. In this case, for the Muller-Hitland agar plates, you're essentially taking bacteria from your last week's lab and subculturing them, i.e. taking the swab and placing them into a new plate. No problem. Great question. Guys, do.
There's no
London.
I give you guys 5 minutes to that increase, trying to stay on time. And then maybe with, yeah, I will see, try to keep it on time.
I have races. I mean, it's three times.
Yeah, actually, 2 different. Try to drive. Does anyone have extra sterile swabs? If your group has any extra, group 1, 3, 4, 5, 6, if any groups have extra cotton swabs, there seems to be a shortage of them. Try not to overuse because technically we had a fresh one today, but it seems like if Swabs. Look at the extra swabs. Reminding me which groups those in the. Let me see. Don't worry, but I'll try to give you extras. Any other groups that need extra cotton swabs? Actually, you know what? I'll leave it. Exactly. So you guys should go to the middle of this bench, and you guys should go to each side if you guys need extra cotton swab. So generation time assignment. Let me just pause. I want to digest. Take a breather. Let's see. I'm gonna go back. So folks, some of you are asking a bit more about the lab assignment generation time due next week. And I would just want to highlight a few things. I talked about this before, but you are, like you're doing for today, for your presentation, having an intro, results, and discussion component or section. And the idea is that you want to be able to showcase two different techniques with two different temperatures and how you're able to measure or determine the generation time of E. coli. To start off, try to, as a helpful point, be as descriptive as possible, especially for your results. And I just want to highlight, at least for your results, that remember you're doing this on semi-log paper that you need to print out, and you're pretty much following. Actually, I think it's better on the slide itself. You'll notice that the increments go up, but they, as you should know, on semi-log paper go up at a multiple of 10. So it'd be like for the absorbance values from the microplate reader, you could start off here and just essentially relabel the axes or the units to show 0.01, 0.1, and then 1, which is something that you may see like here as an example. Whereas starting off, and this is, by the way, for both temperatures, 30 and 37 degrees. And then for, actually, I think I have to go back so that in case some of you maybe were absent, just to really hit this home and give you guys a good understanding of this. You have the values on Quercus, right? So you talk first off your intro, 3 to 5 sentences. Give me a bit to know about what generation time is, why is it important, reflect about population density, the different phases of coli. And then obviously go to your research question and provide specific details there. You thereafter, in your results, will have that semi-log paper. Before you go there, you need to be able to essentially plot the values that you see here across the results. What I mean by the results, look at read 2. This is an example for you guys. Look all the way down up to time point 9. Time point 9 essentially is 2 minutes, 2 hours and 14 minutes. The guide i.e. on on the Word doc provides this information for you. So you should be able to essentially derive from the columns that were shown on Quercus, or on Excel. You would already just copy and paste these values for every time point, and then calculate the following data from the columns to come. So, for example, the mean absorbance values, OD 550, that is corrected. Bear that in mind for your units and for your labels for the axes. You're taking the mean of these 3 reps, right? Which reflect columns 2 to 4 on Excel. Thereafter, you're correcting the mean absorbance, this column or this value with the blank. So you're correcting this across your groups,
and then you're getting your lab average value for each time point. And you're doing that across time from 0 to 120 minutes. I provided those details on the top for instructions for you. But you want to do that and then essentially plot those values and repeat on semi-log paper, and then repeat that process. Before you repeat that process, determine the slope. So you're going to make a curve, right? You correct the values with a blank. So there's a blank. Let's say if we look here, this column is your blank column. So if you, let's do, okay, I don't want to waste time. That's the thing for our presentations, but you essentially take the reps here, determine the mean, place it here as column 5 for all of the groups, then subtract by the blank, column 1. So do column 5 minus column 1 into column 6. And then column 7 is pretty much your last one, which is the average of all of the values from column 6 for each group. And so once you're done that, you do that for all time points. You plot those values on logarithmic semi-log paper. Bear in mind, you have to pay heed or pay close attention, because the values here, folks, are like, for example, I just want you to focus on the cursor, 0.01, 0.1, and then 1. You know, so folks, to bear this in mind, for example, and you need to pay attention, right? Because I don't want to sound like a stickler, but or like I want you to be critical, folks, because the increments here are a certain range. There are about 20 or 30 different sub-increments, the small increments, meaning that this is at 0.01 and this is 0.1, meaning you're going to go up essentially by 0.1 each time, but you need to know how many squares that fits in. And you'll be able to fit it in, but I just want you to pay close attention to that, because that influences where you plot your values on the y-axis. The time points are relatively fixed, whether every 15 minutes for the microplate meter is being provided now. But essentially, you plot those values, you take the slope from your curve, and you make it like, you kind of want on PowerPoint, but trying to make the slope, and then derive from the slope, using the formula above here, what the slope is in log, and then divide 0.3 by your slope to determine generation time. Generation time, your values should be between 20 to 80 minutes. So if your calculations are showing me 0.02 minutes or 80, let's say 20 hours, I've seen this before, then you likely did something wrong. I would say like the range is, okay, the actual range, what you expect to see is 20 minutes. That's your theoretical for 37 degrees. But you can look at literature to see if there's a specific range that you can go over or go off to support your conclusion. But I would find it okay if your results are between 15 to 75. So there is a bit of a range, and I know many of you may be around like the 40, 50 minutes mark. But I just want you to bear that in mind when you're doing the slope. Use the formula here. You could also use the doubling of the y value and then just look at the generation time to compare what directly your generation time is. For your temperature across both techniques, microplate and Bible counts, and the same applies to the other group, the other practical. I just want to say before I answer a few more questions, Bible counts, the increments are different. Bear that in mind. So even though you may start your next graph here at the top half, that's perfectly okay. Just bear in mind that the increments, the units are different, that this is like 10 to the power of 5, 10 to the power of 6, 10 to the power of 7, 10 to the power of 8, etc. Because your range is likely going to vary and be much more larger. So just bear that in mind. It may even go down. I would say go with plotting accurately the results, and that's why the increments, knowing what's in between, is also very important. And if you find Like the sooner, if you have questions, let me know. It's just a bit tricky to do online. But I just want to like go over that discussion. There was another point, viable counts, units. Make sure you have a figure title, make it descriptive for the values I said thus far. For practical 7, their results should be posted already. If you do not find their viable counts or their generation, and I already posted the generation time on Quercus, viable counts on a Quercus inbox. If you don't find their results, use practical 1 or any other practical, any other odd practical, because they would have it at 30 degrees. And then you should be able to see the results and do the same, follow the guide and the steps that I've just said now from before in our previous weeks, and hopefully, inshallah, on the guide. And I want you guys to excel. I want you guys to, all of you to get 4.0s in this course. And I'm technically not allowed to say that, but you guys really, you're provided so much intel information much in advance, sometimes more in advance than other classes. And I want you guys to, get that 4.0. I want you guys to become medical practitioners, doctors, surgeons, wherever you want to be. But you guys have to put that effort now, and hopefully you'll see the fruits come in soon. So bear that in mind, folks. Yeah, do your best. Leave the rest with the best is what I'd say. Anything else? Yes, in conclusions, talk a bit more though about in that concluding paragraph, what you see here and related to what you would have expected. You don't have to put my range because that's my marker range, but for you guys, look at the literature, see what you're able to see. And it's okay if you put in-text citations without a reference. If you can put a reference, that's amazing. But essentially do that on your semi-log paper on the other side. So let's say this is your semi-log paper. You're going to have a figure title, units, very descriptive. You can even separate it for each task. Figure title on the bottom, figure title on the top, label with the legend that's 37 degrees, 30 degrees. Actually, this is a good idea, if you don't mind. Perfect. I really appreciate it, Alta. So I will say, though, as a recommendation, essentially, you can do it like this, but what I recommend actually instead is having them together for 30 and 37 degrees. The temperatures should be on the same graph. You just label them as 2 different curves, one maybe in black and one in red or blue. And then the idea is that you're going to, just like how Alta has done, separate them for viable counts and absorbance values. Make sure that you have units, that the time points are reflective, right? Because they're different. Viable counts every 30 minutes up to 90, or actually 120, 0, 30, 60, 90 minutes. And then microplate absorbance values are up to 120 minutes. So bear that in mind. Very good, very good. So she already has a discussion and objectives. And I just would like to say reorient it so that way you're able to accordingly have the intro at top, discussion in the bottom, and make sure you have calculations of your results. So using the formula that I showed earlier, use that formula, find out the slope, divide to determine generation time. It's equal to 0.3 divided by that slope, and make sure you have units as well. I need to see the units, right? Because after all, you're future scientists, practitioners in medicine, law, physics, wherever you may be. So you need to have units, right? So bear those details in mind. Any final questions before we get started with our presentation? Any questions for the generation time assignment? It'll be due next week at the beginning of our lab. Bear that in mind. So you need to have it present with you. Yes. Intro, pretty much what you have there. 3 to 5 sentences talking about. So talk about. I'll say some ideas, some terms. Okay. I'll just say them now. Generation time. You want to talk about. Pretty much what you see actually in your lab manual, because you already have an intro. So you'll be able to see it. Pretty much generation time, population, populated growth patterns, and then your research question. And be specific, folks. Mention E. coli, mention the temperatures, mention the techniques, microplate reader, and the viable counts. Answers your question, Gordon? Perfect. So now I'm just gonna briefly go to your presentation. So you guys have 5 minutes as a group. If you go up to 6 minutes, it's okay. But I just want us to be proficient for timing. Try to talk about the main things I've shown earlier here. Once again, your intro for each. So if you guys are groups of 3 to 4, the ones of those amongst you that are more proficient and are able to know what they did for a particular exercise can deal with that one if you're in a group of 3. So like the idea is that let's say you're a group of 4. Okay. Antimicrobial susceptibility test and the germicide test result. You want to be able to split that into your groups or in pairs or in trios and accordingly be able to present your intro, results, discussion. Folks, I need to make sure that especially when people are presenting, like as I speak right now, make sure you're not talking on top of someone else. Very important for your participation and for your notes because as I talk about the germicide results, you want to be able to record those details for your post lab. And similarly for the antimicrobial susceptibility test, whichever results you find that are comparable with yours, and make sure that you have a figure caption and figure for your results. I'm going to be asking questions as well, so bear that in mind. You can also write those questions down or write those responses. And what I'm really trying to assess from you guys, and it is your first presentation, so I understand it's maybe difficult, but at the same time, try to be able to see what you can provide, deliver to the class, and be able to interpret and answer questions that I may have for you. Any questions, folks? I'll give you like a minute. Oh, yes. I said this on the inbox. I said this on purpose. Yeah. I did. Should I show it to you? I don't, but this was in March 13th. No, I sent it. I actually sent it at the top of my message. So I said, I understand, or in case, because I sent this before other TAs, because I've taken this course before in TAing, so I know a bit of the material. And that's why I wanted to let you guys know this in advance. But compared to other classes, you guys have more time to prepare. So, how about I give you, if you need an extra 5? Yes.
Yeah, you can specify oral presentations in the next week.
How would you do a presentation without it being oral?
I think it's like one of the teachers' like careers pick up.
No, but I placed it with the reflective practices, so that is part of your your work. And plus, actually, by the way, you'll see it in your lab manual. It's also mentioned in the lab manual, so have a look there. It depends. It may be 2. That's actually interesting. But you wouldn't want to lose marks, right? So what I will say is, folks, in the next 3 to 4 minutes, try to run through with your group any final questions. And that's the time. Now we're here. No, I said this at the beginning of the lab as well. And it's also in the manual. By the way, it's in the manual. Have a look in the manual. You should be able to see it as well.
I think this part is really how much of a difference is something significant difference
Currently, right now, it's very significant. I would just say that if you're able to create zone information and what that signifies, it's great that you have the measurements. It's extremely good for grading, like for yourself to see like the extent of inhibition that exists for a particular antibiotic, for your particular bacteria. But I would say find essentially a summarized like a concise addition as to what you have from your table and provide what your interpretations are. You can't really do stats because you only have one value for each group, for each like antibiotic. So you would have had to do this with like multiple plates. The idea is that you should be able to see the trends and patterns and see what that could imply.
I was gonna say, is our process? Is it such a section? It's like, how do you say it?
Tetracycline. I don't mind, but cycline is actually one.
These measurements, E. coli appear to have a greater jeopardous, meaning that like it was able to inhibit the bacteria like better than it did for S. epidermidis.
Yes, you can. So you could say it's greater, like in terms of zones, that it's greater. Like you can compare as you are now, but staying significantly may be an issue. I would imagine though that it is probably significant for tetracycline between E. coli and Staphylococcus epidermidis. The other ones may be a bit hard to tell. So go with pretty much.
So I think the rest system
The highlight, like, if you actually see a sort of individual or not, it could be if it's present or not in those conditions. For the you can talk about that if you want.
Does that mean E. coli and S. epidermidis aren't the bacteria that penicillin usually inhibits, or
If you conduct it back to your beta-lactamase test, you might actually I'll tell you the results. If you go back, Staphylococcus epidermidis, technically you should see a positive result. So maybe have a look to see, maybe like even what you could do is describe what you see if it matches with what you expect, because technically if you have beta lactamase test, you would be able to know that penicillin based antibiotics have a beta lactam. So accordingly, Staphylococcus epidermidis is positive. You could actually have some. So if you don't see it, maybe just talk about why. But you don't have to focus too much on that. I just want you guys to. I want you to compare between the bacteria. Bacterium. I want you to compare them, and then just provide me intel or an insight as to what that means. No. Hello. It's time to shine, folks. So now is the time for you guys to present. So, may I ask if group 1 will be able to take the stage? If you want to. Wow, this is amazing. Wow, okay. So I said group 1, just a pause, one moment, folks. I said group 1 because technically last on Tuesday I was going in that order. But group 1, do you prefer to go afterwards? No. Why not all of you guys go first? Okay. Let's see. So I'm actually very amazed because well, you guys have more time in comparison to other groups. So what we'll say to make things easier, we'll go with the groups in order. Those that are comfortable, by the way, those that are comfortable that want to go first from that order, they can. If you feel like you need more prep, then we'll go, inshallah, forward to other groups. I want you guys to feel comfortable when you present. And I'm very happy, by the way, very, very happy that you guys were all like competing to be first. That is amazing. Keep up the great work. Now let's go straight ahead. Group 1, are you comfortable with going first? 100%? Let's see. Are you guys 100% comfortable? Let's go. Okay, you guys are ready? Make your way to the front here. You know, probably Dr. Brunson's gonna come and she's gonna be so amazed. What are these? No, don't worry, don't worry. Don't worry. You guys keep it up. And trust me, you guys are doing amazing. Keep up the amazing. Okay. So when you're ready, you can actually, if you want to use the mic, you can. If you want, by the way, as an FYI, folks, I record my practicals in case you guys want to also look on them online. Let me know. If you're uncomfortable with being shown, I'll just tilt down the computer. But generally speaking, if you guys want to use the mic, you can. We may have a wireless mic as well. Who wants to use a wireless mic? You guys want to be professors?
I think I'm loud enough.
Okay, that's okay. If you guys want to, let me know. This is fun. So, yeah, don't worry. You guys are amazing. I'm so happy we're competing and just keep up the same energy. Keep it up.
What's the context?
Keep it up. Okay, when you guys are ready.
Oh yeah. Hello everyone. So we actually had.
Folks, let's pay attention. We all have to listen. Make sure you're not speaking in front of other folks. Let's pay attention. I want to make sure everyone's paying attention. And whenever speakers are speaking to us all, presenting, make sure that you're also speaking with an audible tone, so that way anyone from the other ends can hear you clearly.
So should I use this?
Just speak a bit louder, if you can, or with the mic.
Nah, I'm good.
Okay.
So we actually had two plates. So we had E. coli and then we had the S. epidermis. So we treated them both with 5 antibiotics and then we incubated them overnight. And the goal of the experiment was basically to determine if the antibiotic inhibits the bacterial growth and what path and the bacterial static, or was it either bacteriostatic or was it bactericide? So bacteriostatic being inhibition and bactericide means it would kill them. So
Hello. So, what are the results for the E. coli? All the antibiotics for some inhibition, except penicillin, which shows that it's a bacteriostatic, and for the S. epidermis, 3 antibiotics produce inhibition zones, while penicillin and tetracycline did not. This suggesting they only inhibited, and also shows that the tetracycline can act as the bactericide or the bacteriostatic, depending on factors like concentration, bacterial species, and the other. That's it.
So hello. So basically what this would tell us is that for penicillin, it had a bacteriostatic effect. So what this means is that it was basically because it has E. coli is a gram-negative bacteria. So this means that it had a much thinner peptidoglycan layer, and it also had an additional outer membrane layer, and this acts as a barrier. So this actually makes it difficult for the penicillin to penetrate and reach the peptidoglycan layer. And then this would therefore reduce the effectiveness of the penicillin, and then therefore it'll lead to a bacteriostatic effect. And then for the other one, it was penicillin and tetracycline that were bacteriostatic. And this is actually because for penicillin, many S. epidermis strains produce beta-lactamase, which breaks down the penicillin. And then this makes that unable to kill the bacteria. And then for tetracycline, it's inherently bacteriostatic, and that would prevent the bacterial growth and inhibit the protein synthesis, but it won't kill the bacteria. And then for limitations and extensions, so one limitation we were thinking of saying was for antibiotic resistance. So that would be one limitation, because the E. coli and S. epidermis, they exhibit like antibiotic resistance, and then to common drugs like penicillin and tetracycline. And this would lead to inaccurate interpretations for either susceptibility. So then, for example, like the epidermis, it might not be able to resist the penicillin due to the beta-lactamose production. And then while certain strains like E. coli, they might show resistance to tetracycline, but not penicillin. So this was one limitation. And then one extension that you could possibly use is that we can use a broader antibiotic panel. So then this would expand the panel of antibiotics used to test a variety of classes, for example, such as like aminoglycosides and like macrolicides as well. And this would help us get a better understanding of the bacterial susceptibility and the resistant patterns of them as well. And it would also help identify alternative antibiotics as well. So this is what we came up with.
For green class, think about your column. So this, so everyone else is aware, you only have to focus on your column from the last time point, or even if you have the trend for others, that's okay. They talk about compare pretty much positive all the way down to your highest concentration. When you
So, disinfectants or chemical germicides produced with the intent of removing pathogens, especially from surfaces. And one of the most commonly used household disinfectants is bleach. In this experiment, we tested the germicidal effect of 0.5%, 1.0%, and 4.0% concentration of bleach on gram-negative E. coli. And the effect of the disinfectant will be measured by using a microplate reader to determine absorbance values at 550 nanometers after 15 minutes of incubation. Bacteria which survive the bleach addition will contribute to the turbidity values, resulting in a higher absorbance reading.
With the results, the higher absorbance was observed in the place. For the higher absorbance, which is a plate with more bacterial growth and also absorbance that is same as the positive control, which is E. coli, was observed in the bleach with 0.5% concentration, and the absorbance was 1.39. And the lower absorbance was observed in bleach with the highest concentration, which is 4.4%, which exhibits that this one with higher concentration had the most effect in inhibiting the bacterial growth. So the complete inhibition, which is the absorbance close to the blank or negative control, was in plate F, which is same as bleach with 4%. The actual absorbance, which was calculated by subtracting the blank absorbance from samples, were observed as 0.5% of bleach had a 1.227, 1% bleach had 0.48, and 4% bleach had an actual absorbance of 0.08, which is decreasing in order to the increasing concentration of bleach.
So overall, we found that 0.5% bleach had practically no effect on the growth of E. coli, being almost identical to negative control in absorbance. At 1.0%, the bleach addition reduced the amount of E. coli by over half, whereas in 4.0% bleach, there was almost no E. coli present. This is in line with the accepted results that a higher concentration of the disinfectant will result in a higher proficiency in reducing the bacterial growth. Additionally, this experiment suggests that at low concentrations, disinfectants are ineffective at bacterial control, being unable to get past the cell wall and denature the protein structure within. In household settings, bleach is usually sold between 5 to 9%, which is higher than that of the 4% used in this lab, and suggests that the commercially sold product is effective at reducing E. coli growth.
But here's what we'll do. Talking about the antimicrobial susceptibility test. Amoxicillin is so think about like these sets of questions that are a bit novel for like an lab exam. Amoxicillin is a type of penicillin. It was part of the family of penicillin-based antibiotics. Based on your results for Staphylococcus epidermidis, what do you expect to see following treatment
with amoxicillin.
So since it's a type of penicillin, it will be bacteriostatic because it won't, because the penicillin will penetrate into the layer and it won't kill the bacteria.
But what would you see on the plate?
But what we see,
We won't see a zone of inhibition.
yeah, it would be like there would be no growth, it would be static.
But so if you don't see a zone, you don't see a zone, then it's not necessarily bacteriostatic because unless you see a zone of inhibition, and that indicates that there is antibiotic resistance. So what I would recommend is actually saying that essentially that there isn't a direct effect on the wait, sorry, did you see a zone of inhibition?
Or
Penicillin?
No.
No,
For Staphylococcus aureus?
No.
No? Okay.
that's why.
Other groups, is that the case for you as well? Yeah. Not all of you. Yeah, I think so. For penicillin. Yeah, so okay, so there may be some differences here. But
For both, we
We
did
didn't
not.
do that, you know.
So maybe I would say in your profile, describe because technically you may see these results. Overall, great job. Complete taking notes of other groups. And in the meantime, group two will be going up unless you want another group to go. Okay, okay. Congrats, guys. Great job. Keep it. Just want to highlight, folks, try to be on time. I'll try to give a reminder for a minute, so that way other groups can go in. If it can be done in 4 to 5 minutes, if possible, that would be amazing. Just let me know. I think by the way, the mic is I'm expecting things, but you can use the podium. When you're ready, when you guys are
Yeah, yeah, we'll go first because they just did theirs first. Okay, so overall antibiotics are used to treat bacterial infections. They can be categorized as bacteriostatic, which just means that the bacterial cells won't be able to multiply, or they can be bactericidal, which would be killing all the bacteria. They can also be classified as narrow spectrum, where they are targeted limited groups, and then broad spectrum, where they would target both gram-positive and gram-negative bacteria. And so what we did in this experiment, we did it so that we can determine kind of the level of susceptibility of specific bacteria to the antibiotics, which we then would measure through like observing the diameter of our zone of inhibition around each antibiotic disc on the agar plate. So we use the antibiotics ampicillin, chloramphenicol, penicillin, tetracycline, and streptomycin on both E. coli and S. epidermidis, where we both like heavily inoculated both plates and then placed each of the antibiotic discs on the plate and then incubated it overnight at 37 degrees. And then our results.
So when we took a look at the response to the antibiotics, we saw that S. epidermidis from the largest response to the smallest response, we had chloramphenicol, streptomycin, tetracycline, streptomycin, ampicillin, penicillin, and then tetracycline. And then as for the E. coli, the largest, the smallest response, chloramphenicol, tetracycline, streptomycin, ampicillin, and then penicillin. If we take a look at the strains of antibiotics, we have the ampicillin and penicillin, which are both beta-lactam antibiotics. And if we took a look at our beta-lactamase results from the previous tests that we did last week, we expected a larger response to beta-lactam antibiotics from the S. epidermis, just because it turned pink in color. However, this didn't reflect in our results from last week, as it had a much smaller zone of inhibition than expected. Some limitations, there could have been some sort of breach in aseptic technique when we were inoculating the antibiotics onto the streak plate, or like the other group mentioned, there could have been some sort of antimicrobial resistance for the bacteria that was spread plate. And then as Fauna mentioned, the narrow and broad spectrum antibiotics may have had different effects. As for the beta-lactam antibiotics, they inhibit the PBPs, or the penicillin binding proteins, also known as transpeptidases, in the peptidoglycan wall. So if you compare the gram-negative E. coli and the S. epidermis, which was gram-positive, with their thin and thick peptidic glycan walls respectively, you would expect different responses to the beta-lactam antibiotics, but this wasn't necessarily reflected in our results as they both had smaller zones of inhibition.
And the second experiment that we conducted was the microplating experiment to test the different concentrations of bleach and how they would react to different types of bacteria. So we had the concentration 0.5%, 1%, and 4%, and we used a microplate reader to measure the bacterial growth density, which would show whether the different concentrations of bleach resulted in lower concentrations of bacteria.
And for our results, to get the true absorbance, we subtracted the absorbance we got from the blank, and it resulted from the positive as 1.076, negative being negative 0.008, the low concentration being 1.249, the medium concentration being 1.198, and the highest concentration being negative 0.002, which means that the OD does decrease as the concentration of bleach increases. And as the high concentration is negative, it means it is killing the bacteria.
And possible limitations of this include the fact that the bleach actually causes the bacteria to clump sometimes, which could have disrupted the light and the absorbance readings, which would account for any discrepancies in the absorbance readings.
Since our positive control is low. Medium content.
So I have a question. Yeah.
No questions.
Yeah, I think actually I'll do them at the end with everyone. I mean, it's easy. Great job, you guys. You did that. If you're done, yes. Three, two, one. Are you ready? Let's pay attention, folks.
Previously, we all know that antibiotics are used to kill or inhibit the growth of bacteria, which could be categorized into two sections based on the target spectrum and then the target specificity. The target spectrum, narrow or broad, and target specificity is anti-bacteriostatic and bactericidal. Where bacteriostatic is used to inhibit growth, while bactericidal is used to completely kill the bacteria. So for our experiment, our purpose was to see how vitamins and antiseptic to anticidal could inhibit the growth of E. coli or Staphylococcus epidermidis through a so by doing it this method and speculative technique, we're going to speculate on two the bacteria plus the nutrient outer plate and then use targeting this antibiotic to analyze zones of inhibition.
Okay, so for our results, so we did two experiments, which was the E. coli and then the S. epidermidis. So in our results, we're able to see a drastic difference in the penicillin and the E. coli and in the in comparison to the S. epidermidis. So in the penicillin, there was no zone of inhibition, and this could be explained by E. coli being gram-negative, and also due to its outer membrane barrier, and due to penicillin being narrow spectrum, you only target a limited group of bacteria, and also in comparison to the other antibiotic, which was CHO, you can see that it's broad spectrum and was able to kill both gram-negative and gram-positive in comparison to penicillin, which only killed the gram-positive one. And also in our results, we're able to see that some circles were jagged, and this could also be explained by
bacteria not getting enough chance to properly absorb into the antibiotic disc. And the limitations in this result would be aseptic techniques or some cross contamination. And for the penicillin and the E. coli, that could be explained by some antibiotic resistance.
So regarding chemical germicides, I don't know it's particular in the growth, which could be categorized into disinfectants and antiseptics. So the purpose of our lab was to see how the varying concentrations of antiseptics and disinfectants affect the growth or kill bacteria cells. We diluted the bacteria and mixed it with some disinfectant or antiseptic of interest, and let the mixture settle for 15 minutes, and analyze absorbance of the mixture to analyze turbidity to see if the bacteria cells are growing or not after the applying disinfectant. Regarding the results.
Yeah, so regarding the results, we can see on average we're column 3. This was low concentration, medium concentration, high concentration. We saw the opposite of what we expected, which is kind of sad. I'm to blame. But essentially in the low concentration, you see a low absorbance, meaning there's little coli present, and increasing to a high concentration of hydrogen peroxide. So this is partially expected that, actually this is the opposite. So E. coli came negative for the catalase last week. I mean, they had a bit of activity, but not a lot. So we would have actually expected them to be susceptible to H2O2 because they wouldn't be able to break it down, the catalase enzyme. So some limitations of this could involve microplating techniques. So when discarding a supernatant or the disinfectant, could have been that we didn't get rid of enough or too little, creating contamination. When micropipetting, occasionally a pellet might have gotten sucked up too, which would have disrupted the coli count. And to extend this next time, I would say being more careful with micropipetting techniques would be great, a great extension, and doing multiple replicates rather than just one straight replicate. And yeah, overall, I mean, it signifies that H2O2 isn't a good disinfectant towards E. coli, but this could be different according to the literature.
Any questions for me? You can actually repeat now. Move forward, you're up next. If you guys can keep a track of time, there's also the computer there that maybe is on. Yeah, you guys should see the time accordingly. I also have a timer here, but go as soon as you can.
So this swab is basically about how we can stop bacteria from growing using germ-killing chemicals. And we use 2 specific types, which are disinfectants, which are used on surfaces, and antiseptics, which are safe to use on the skin. So we would have seen how well these chemicals work against 2 main types of bacteria, which are the gram-positive and the gram-negative. And to do this, we use the use dilution test. And then another test we will do is the catalase test, which helps to tell 2 bacteria apart, which are Staphylococcus, which makes an enzyme called catalase. So when we add it, hydrogen peroxide, it bubbles. And the second one is Staphylococcus, which does not make catalase, so there will be no bubbles. So we'll also test how bacteria reacts to antibiotics using the disc diffusion method. And this basically means placing antibiotic soap discs onto the bacteria and seeing if they stop growing. Some of the antibiotics we tested included penicillin and ampicillin, which attack the bacteria's outer wall, and then chloramphenicol, and then tetracycline and streptomycin, which stop bacteria from making proteins. And basically, a big question we asked is, do different antibiotics work better on different types of bacteria? And so our group, we were testing the hydrogen peroxide, specifically the Staphylococcus epidermidis, and our results.
Indicates antibiotic sensitivity. So when it came to our results, some of the conditions in coli for all antibiotics was generally larger than it was for Assaculomitis. This is especially true for tetracycline,
which was able to inhibit E. coli better
than it did for encephalitis, as it had a greater zone of inhibition diameter. So basically, this means a larger zone of inhibition for E. coli showed that there's less resistance to the antibiotic. When it came to ampicillin, cephtromycin, and chloramfenicol, they all generally had a similar size zone of inhibition for both E. coli and S. epidermidis. But when it came to penicillin, there appeared to be no zone of inhibition for both E. coli and S. epidermidis. This was expected for E. coli, but is unexpected for S. epidermidis because penicillin is bactericidal, meaning it kills bacteria. So for S. epidermidis, we know that penicillin should be able to inhibit so for S. epidermidis, we know that penicillin should be able to inhibit that. There should be a zone of inhibition because of the causal reading we did on the beta-lactamase test. And our discussion. So, as she mentioned, all of them showed a zone of inhibition except for penicillin, which showed this, which is, as I think everyone said it, it's because E. coli has an outer membrane that prevents penicillin from reaching its target, and due to its narrow spectrum nature, it only targets limited groups. So that may be a reason why. And although there was no zone, but there was a slight bit of weaker zone that we noticed for S. epidermidis, which may produce beta-lactamase enzymes that break down penicillin in general. So not that it was a clear zone, but it was, we did see it in our results at least. So for us, tetracycline had the highest, the largest zone of inhibition for both E. coli and epidermidis. One reason can be because E. coli is gram-negative and tetracycline is highly effective against gram-negative bacteria, as it can pass through their outer membranes using foreign channels. For us, yeah, the biggest thing we would say that did not get us the results we expected was a few limitations, such as the uneven bacterial spreading. So I think we had pipetting problems where we suspend the bacteria and pour it in. That might be due to because each one of us did the spreading, it kind of came out different depending on who did it. That might be a reason why. And because the antibiotics diffuse at different rates in the agar, that can also result in some showing signs of zone of inhibition, whereas others don't. So to improve next time, I guess, let one person do both of the streak plating, let one person just do the pipetting to ensure that whatever is done is done.
Okay. Yes. Okay. For the disinfectants, we were for hydrogen peroxide against epidermis. Okay. Sorry, I'll be quick about this. Okay, so I'll just talk in general trends. Trust me on this when I talk about the 2 hours and 15 minutes. So for blank and for negative control, we were basically near consistent against all the time, and they are relatively close to each other, so probably no contamination. For our positive control, there was a gradual decrease for every sample, including the disinfectants we were testing, for decreasing absorbance. This is probably because we started with an OD of 0.85, so probably too high. And we're starting near death phase or station phase. But this doesn't generally consider, this is not considered effective the disinfectant itself. In fact, they were generally close to the positive control, so it's probably didn't have any effect as expected since these are calibrated. So in turn, also limitation fundamentally about this is essentially we're using a big drop in absorbance as a proxy for lysis for bactericidal activity of our disinfectant. If our disinfectant was able to kill our bacteria without causing lysis, we wouldn't be able to differentiate between, well, if they're viable or not. Also, another fundamental limitation, this is testing for specifically bactericidal effects of the disinfectant, not bacteriostatic. We could possibly make improvements for this by starting with a lower OD and also like in tandem making some new samples to also coincide measure or disinfect at the same concentrations we were using before. Anything?
Going. So try to take 5 minutes post. I may have to go a bit over in some, like the last group may have to go a bit over. Please try to, be patient accordingly. With daylight savings, likely you haven't been too gravely impacted for some of the courses, but let me know nonetheless. You guys can know. Okay.
I have a quick question.
Yes.
I have biochemistry after this.
Can you speak?
Are you ready to go first? Yeah, can I go first? Yeah, okay.
Okay, so antibiotics are chemicals that can inhibit the growth of or kill bacteria. They're used to treat bacterial infections. They can be bacteriostatic, where they inhibit growth, or bactericidal, where they actually kill the bacteria. And they use many different mechanisms to do so. They can be classified as narrow spectrum, where they only affect a certain few type of bacteria, or broad spectrum, where they can affect both gram-positive and gram-negative bacteria. And in this experiment, we used the disc diffusion method to inoculate 2 different nutrient agar plates with either E. coli in one plate or S. epidermidis in the other plate. And we inserted 5 different antibiotic paper discs to test how susceptible or resistant these bacteria are to these different antibiotics, incubated them overnight at 37 degrees, and then following experiment day, we looked at the zones of inhibition around the discs to assess. And our research question was, how does the size of the zone of inhibition correlate with the effectiveness of different antibiotics in inhibiting bacterial growth? And can this be used as a reliable indicator of antibiotic susceptibility in both E. coli and S. epidermidis bacteria?
In discussing results of the chemical germicide tests on E. coli and S. epidermis, it appears that E. coli exhibited resistance to only to penicillin, while S. epidermis showed resistance to tetracycline. And for E. coli, we saw on ampicillin and streptomycin, the jagged, not killing but inhibiting growth tendency. And on S. epidermis, we saw on penicillin, ampicillin, and streptomycin, same thing. The bacteria had jagged inhibition in growth, and E. coli and penicillin resistance. The resistance of E. coli to penicillin is consistent with the known mechanisms of antibiotics resistance. Many strains of E. coli have developed resistance to the production of the beta-lactamases, as mentioned on the first group, which hydrolyze beta-lactam ring of penicillin, rendering the ineffective prevalence of plasmids that carry resistance genes among E. coli populations in various environments. For S. epidermis and tetracycline resistance, the resistance of S. epidermis to tetracycline can be explained by the presence of efflux pumps that actively remove tetracycline from bacterial cells, thereby reducing the effective concentration. Errors and considerations. So we discussed, and we found that testing conditions could go wrong, and aseptic techniques can be a result of error, and interpretation of results. That's you want to add
any extensions that we could do to the experiment is looking at varying concentrations of antibiotics, different combos of antibiotics, or even looking at the effect of different environments, like different pHs or temperatures on the antibiotics and how the bacteria react to them in those situations.
Okay, so for the first exercise, we actually investigated the effect of chemical germicides on microbial growth. And there's basically chemical germicides divided into 2 components. It's antiseptics and it's disinfectants. The main difference is disinfectants affect surfaces, non-living surfaces, so surfaces over the table or anything. And antiseptics actually affect the living tissue itself. And what we were trying to analyze or I see was the difference in, or the change in concentration and how that would affect the overall microbial growth. And the way that we would test this was by measuring the difference in absorbance levels across those concentrations. And so our group, which is group 5, we had E. coli, and our germicide, or specifically our disinfectant, was ethanol. So we measured at 25%, 50%, and 70%. And Jacob will.
All flat, and then we can see that while the control is correct, we saw that there was a C, there was nothing, it's correct, and then for E, it's 25%, and then 50% and 70%. So we see that there was growth at low concentrations and at medium concentrations, but then it was significantly reduced at 70%. And then what this basically says is that at high concentrations, ethanol is pretty effective at killing E. coli, but then there was significant similarities between both 25% and 50%. And for the limitations, we basically are thinking that there was improper pipetting, and then maybe there was a possibility that it wasn't actually 50% and that the concentrations were similar. And then for our future extensions, we're thinking that because these values are so similar, that maybe it's something between 60% ethanol that might be in between these values. And then wrapping this up, it's kind of important to do this experiment because it's important in medicine to determine the resistance for germicides and bacteria.
You guys ready? I know it's a bit tight for time, but if you're able to do just the germicides, that's okay. If you want to do both, I don't want to interrupt your delegated tasks.
Let's just do the germicides.
Okay. Whenever you're ready.
So for our first activity, we wanted to test the effect of specific germicides on in our case, S. epidermidis, which is a positive gram bacteria. And to perform this, we measured the OD at 550 nanometers and at different concentrations of a specific germicide. In our case, it's ethanol. So we have a dilution test, which is 25% ethanol to 50% ethanol, and then 70% ethanol. And we measured the OD of the when we inoculate each condition. We measured the OD at 550 nanometers to determine the growth of bacteria under these conditions.
In group 6, you can see that 25% ethanol and 50% ethanol doesn't really have a great effect on the inhibition of bacterial growth of Staphylococcus epidermidis because it is close to the initial absorbance value, which is 0.85. But when we see the 70% ethanol results for the absorbance, when you minus with the blank value, it shows that it has a higher effect on the inhibition of bacteria. And that is because ethanol in itself is a disinfectant and it causes denaturation of the proteins of this bacterial cells, causing the cells to not be able to survive properly and be killed, essentially. And yeah, so that's the conclusion. And for the limitations, it's not shown here, but for the time interval at 105 minutes, we saw an increase of the absorbance for the 50% ethanol, which is not expected. So we can say that possibly more of the cells in the dilution tube was added to the further dilution too, which explains possibly that limitation. And for the extension, because we only tested for the effect of the disinfected ethanol, we would like to also test for other types of bacterial cells, such as those of spore-forming bacterial cells, and see how the concentrations of ethanol affects those types of bacteria for future research.
We conclude that. So at higher concentrations, ethanol did have an effect on inhibiting the growth of bacteria due to the denaturing of the bacteria itself. It can also be mentioned that there can be tests done at higher concentrations to further see this effect, but at higher concentrations it does seem to have an effect, but at low concentrations it does not inhibit growth as successful.
So this shows that sterilizing your benchtop with 70% ethanol is effective in preventing cross-contamination.
Great job. Well done. Okay. So I'm just going to go up front and just highlight in summary. I won't, to make it easier for you, I'll just be mentioning a few questions. You already have a few of them already included for the reflective practice. So if you haven't done them for the last slide of Lab 7, then inshallah have a look there because they are questions that you guys want to answer. I do want to highlight though, so one, in terms of presentation style, you guys are amazing, very engaging indeed. I was so happy. It's actually like a very good highlight that you guys were eager to go first. So I adore that and all the best. Keep up the amazing work. In terms of a few pointers, always make sure that you're specific with details that you provide. For example, when you're doing your intro, when it comes to the specific species of bacterium that you're looking at. In FLRs, you want to start off with the full name of the genus. And I know your presentations, you don't necessarily have to, but start off with saying, like, for an FLR immune generation consignment, Staphylococcus epidermidis. And then you can, thereafter in italics for all of them, say S. epidermidis, etc. Then when it comes to in terms of other things, maybe just to highlight your presentations. So this one that's highlighted right now is the one that you would actually be using for your data analysis. And some of you are looking down here, but because of it being reflective after 2 hours of incubation, there's a few things that you can keep in mind. One is the results, the trends that we talked about, and also the limitations that are also, that are quite selective. So I want you to also notice that in the microplate reader, it's at 37 degrees, but it's also shaking, meaning that essentially the idea is, and the hope is that you will be maintaining that resuspension of the bacterium, and such that it will be left solid. The concern that could occur is that if there's a peak at a given point, or if there's an anomaly at a specific point, it could be a miscalculation from or misinterpretation of the absorbance reading from the microplate reader, because at the end of the day, it is reasonably UV that are passing through 560 nanometers to be able to fit this. Visible light, 550 nanometers. But nonetheless, so you guys usually talk about the different trends that you see. Some of them are being different from what you would have expected. So you can highlight the limitations indeed, yes, but there's also the confounding implications of, like, from the germicides that we looked at, from bleach. Think about like your future laundry detergent. What types of bleach would you use, let's say, for your lab coat, for your concentrations, right? You'd go probably over 5%. Now, in terms of comparing like Staphylococcus epidermidis versus E. coli, these are things that we've touched upon. Many of our beloved students have also shown it here. I'm talking about you guys in third person, but essentially the idea is that you guys want to be able to articulate this in comparing both sides of the story. Likewise for the antimicrobial susceptibility test, and importantly, know the modes of action. Some of you have pointed this out. For example, streptomycin is an inhibitor for protein synthesis during translation. So these are things that you want to talk about and their potential effects thereafter for the specific species of bacteria. Pellets. Some of you may have also talked about the pellets in use. Streptomycin, tetracycline. Tetracycline likely, I'm just going to end off with a few final points. Tetracycline, you would have expected to see different results for Staphylococcus epidermidis versus E. coli, and the same for penicillin. Try to see and know in your post-lab the differences between the two and how important the other tests come in, like the catalase tests for the germicide of hydrogen peroxide to the cephanase or beta-lactamase test when it comes to the penicillin family or beta-lactam antibiotics. That includes ampicillin, penicillin, and other members of the penicillin family like amoxicillin, azithromycin, etc. So think about those concepts. A lot of this is replicable to and applicable to your future practicals, post labs, and importantly for your lab exam. So with that, any final questions, folks? Generation time, assignments, presentations, anything, everything's okay? So I wanted to summarize. I summarized. I don't mind actually, but I summarized those questions in my final concluding statements for today. So think about those questions that I talked about. Think about the commercial use of, let's say, bleach, for example, or finding out the modes of action within antibiotics. Those are things that you want to highlight. I wanted to save time, but all the best, you guys. Amazing, great job. Keep up the amazing work. And just try to stick with timing, especially for the presentations, wherever you can. But besides that, great job. If you have any questions, let me know. All the best, everyone. And looking forward to seeing you in our final set of classes. Incubating. Oh, great question. Incubating your plates for exercise 1. You're going to put them in the bottom incubator at the top left side. For exercise 2, they need to be arranged at the fluorescent lights there, or at the light switches down that side, with the aluminum foil wrapped up and labeled for your dark treated plates, dark incubated plates. And the ones that are on light, arrange them in rows and label them accordingly. For any other plates, let me know if you want to keep them. And yep. And make sure to clean up, of course, your benches, sterilize them accordingly, and return anything that was unused to their appropriate locations. Into the middle white tree.
We're not supposed to wrap this
for exercise 2.
With the light middle.
No, that's not the lighter.
This is the
That's the lighter.
Oh, you've already done it.
Light dark is exercise 2.
Oh, this is 1.
Yeah. Are you sure though? I just want to make sure.
Yeah.
Exercise 1 in the bottom incubator, top left side. The same, actually, in the same location. Yes.
15 That was number 1, I'm pretty sure. 15 minutes of.
Yes. Yeah.
Nakansaki's on 2
Yes. So don't worry about the last tech is responsible for testing. Don't worry too much about that. Lab exam. Lab exam is part of your final exam. So it's going to be incorporated likely at the very end of your lab exam. Your final exam, it'll be part of your booklet, your question answer booklet. I don't necessarily know. You'll have short answers, but we'll see. Yes. Yeah. Remind me. No problem, Hannah. Did I answer your questions? If you have more, let me know. You too.
And then like this is this was the declaration for this.
Okay, you'll include that.
Yeah, from the.
Okay, perfect. Great job. You can include those for your post lab for next week. Apart from that, great job. Any questions?
I guess I do have. So when you explaining the assignment, I actually did not get labels.
For the generation time?
Yeah, for the generation time.
Okay.
In terms of like the graphing, I don't understand.
Okay.
Yeah.
Let's see. I don't have a paper example of it, but if you look at the slides and look at my guide, I try to make it easier to understand. So you have semi-log paper. You're going to include the intro, results, and discussions on the other side. The results include your calculations from the slope of the curves that you have. You'll be making 4 curves, which will be on the front side of the semi-log paper. And this is all what I did is that you'll have a section for upper counts, or for the microplate results. Viable counts I already posted on purpose to make it easier for you guys. You don't have to do as much data, like much calculations or many calculations. But the point is that if I look, yeah, it's also shown here in the, sorry.
So the data we're using, like in terms of the X and Y.
Yes.
Like which one X?
So on, I'll show you. So first of all, viable count or so this is going to be in time. You make 2 separate graphs. One is for viable counts, one for microplate results, and you're going to have 2 curves for each. No. Okay, no problem. In the meantime, any other questions?
Question, for generation time, do you mind if we fix our data, as in like we remove outliers and stuff when we present this?
No. It depends how many outliers. Well, how many outliers?
I haven't done the calculations yet. I'm just checking like statistical analysis. Like, do you mind if I remove outliers?
I doubt you will find out. If you like, are you seeing for your slope?
Yeah.
So are you feeling this show out an example? This may not work. So let's say this is your okay. Take care. Make stuff around how you do your curve, for example. Let's see how the point is. I don't mind if you consider it as an outlier. I don't use it as an outlier. If you try to say a second one, go for number. It'll simply balance, you know, the two. So essentially, it's calculated into a better effect from the outlier here, the outlier that's there. That essentially has an even distance such that when you put the flow, both of them should have roughly equal. It's actually more, don't make it arbitrary, but you would primarily focus on your log, in your life. You would want to make your flow essentially linear. And to an extent, it's not always going to reflect the residuals maximally as you're doing it. But at the same time, try to essentially account for outliers as much as possible, be as close as possible to them.
Okay, technically, since we're using aggregated class data, technically, doesn't it really look like this?
No, so because you're doing the mean.
No, when I'm doing the mean, if the mean is like shifted into like box plotting, box here,
yeah, I understand.
Here, like you might have like.
I'll be honest with you, not required to use box plotting. That is the main that you appreciate. But technically, with the goal, so technically in case of generation study, what they really just want us to show is the main values of plots, not necessarily showing the ranges, variation, or distribution of data from across groups, just pretty much the lab mean value of all groups for each time.
Okay, so even with that in mind, okay, if I just represent the other those curves, do you mind if I fix my mean detected by a loss?
How would you fix
more accurate, just basically just remove it from my mean calculation and whatever.
Technically, no. I don't think you will actually find it to be a grave find a grave anomaly. If I find a grave anomaly, then I will. No, I don't think. And plus, the other thing is that these are only, if you find an outlier, probably out of like 3 different time points out of the 12, out of the 9 that you have. And out of those 3 time points, you may only find 1 or 2 of the class data points that are actually present. So if you think about it in the large scale things, you have maybe 6, maybe we'll see even 3 different outliers out of a list of probably 90, 9 time points out of 54. So it's a very small margin. Like you're going under 5.
But technically, aren't I accounting for this for each time? So I'm technically making a profit every time.
Yes, but because you're summarized for the 6 points, technically speaking, so the idea is that I know there may be some outliers, but at the same time, I doubt that they will make a significant difference in the greater than percentage of our breed. For example, let's say you find revalue which is generation type, whereas following the I know that's some it depends on one size. In the case of I'm not going to be organizing potentially. That's why I would actually prefer essentially use all of the data and if you're correcting for, I know we have like, that's another thing. We also have 3 different maps for that. So when you really think about it, aggregating it all together, over 100 values, you're only having 5 different potential. I haven't really I don't think there would be major outliers. So even if there was a case, I mean, honestly, I don't think we need the outlier. But nonetheless, I doubt it will impact your attention, especially this large rate, which is 15, sorry, 16 minutes. Not to I wouldn't expect you to essentially, like, I think I would recommend in general, keep the data as it is, but like take the averages correct, and then correct accordingly. Thereafter, essentially, allow and accordingly those values will be corrected for each time. And by the way, another thing is that earlier endpoints are not always necessarily to be like fixated within the small range, meaning that it may not drastically change in each time slowly, which is slightly tangent. But nonetheless, great question.
Second question, so did just a question about my analysis seem probable good?
It was good. I think, and I didn't want to because I was worried that.
No, I know, I get it. You're worried by other people.
But nonetheless, I actually appreciate I keep it up. If you find that, let's say there's maintain the engagement of the crowd. That's what you already did. I'm happy with your. Actually, I would say no. Your performance is not to check on. Yes.
Do you have a separation between your lab and the other lab?
Yes, they were. There was a sep.
You put everything like that. Okay. Most of these are wrong. Yes. Now, you really need to teach your lab on time. Okay. Yeah, that's the argument by default.
Yeah, the bottom comes from literally. In a sense, is like everything's on itself.
So, folks, if you have more questions, give me a bit of time. I do have to.
I'm not going to just focus on another thing, right? Yeah, you got it.
It's the same thing. You're just falling into the thought. So all it is like versus top honesty, second degree, small honesty. If you separate them, they're outside. And then another thing. Why do you have there's zero.
Yeah, what do you need?
One space.
I use them to
but that's when you come by 0.0155 or 0.01. Obviously, here's 0.2 for each one.
So in terms of usually if you gain access to
when that happens, like, so you're gonna have so for instance, time at successfully 3 months into 0, so and then you ask how to be absorbed and how to do that. There's a lot of that actually just
Yes.
oh, you said the question that this was 0.01 so you might scale this.
Yes, yes.
This one,
Oh, okay, okay. If you could, so technically you have a line of best. Oh, give me one more.
Question, for correcting the blank Is it better to correct each point before I take me, or should I, is it how I get to take the blank off the price?
It would be better, different meanings. Reason being is if you actually do it for all of them, it'll be the same reason. But generally speaking, it will still be ideal to do it. So we have taking the mean first. Then correcting the top. Before I want to
So you were describing what was supposed to be on the graph, right? That variable class and then the which have absorbance readings and they're supposed to be like 4.
So instead of 4, I actually recommend
I was confused on how that to.
So I think we should open that. Or if you have a look,
I have like a manual population.
that would be good.
I have the user knowledge. Like this is the mean for all. Like the graph is 6. Column 1 would have been blank, and column 6. Sorry. Column 1 is a blank, and columns 2 through 4 are your replicates. So you're going to average the replicates for each of them. And then you subtract the blank from the mean, and you get your absorbance, and you just average that.
So like, if you look at the guide on the window, then you can be able to see that it's actually pretty much opposites. We have the column 2 to 4, and the right result is often the result on the word one, and then we do the calculation. Then calculate the now average from that.
So we're just back in the
The average that is corrected for all groups, all components, for each sample. And then the thing is when usually how approaches the graph. If you want to see the graph, will have from one end to that the other, essentially in two groups for one group, and then you have a separate graph, or you can do the on one half and you do the other. But essentially you'll have for each technique a separate graph. And it could be within this finite range, or it could be like just how it's shown.
Yeah, I think the curves, there's sometimes a circuit draw curve than tangent, but the top is.
That's okay. Both ways work actually.
So this is fine.
Actually, we can see as line when we so it's okay. It's not nothing we can do. You don't really see a curve because I don't think on a arithmetic scale, so like not even the 54.5%. But in this case, you're going up by multiples. So in that regard, it is exponential still. However, you're actually seeing it log as a linear method.
And also there was a question that was asking, how this has 0 or 0
So the y-axis you can do non-vision variable, so actually
it's not like binary. It's like double, but goes to the next values.
no. So 0.1 is actually technically you can do both, but the issue is that you yeah, I don't But how do you have it really? And your calculation, they were.
I got 7.
That's okay. That's okay. I would say, let's see. I would recommend doing it, doing the way that I presented it. Essentially, technically are going up by multiples, but essentially that range is outsized. So pretty much we've just expanded it.
It's like zooming into the
Exactly, exactly. So it's essentially it's okay. Follow up with 0.0, start off with 0.01 and go up a little bit, but make sure that you're following the log scale. Essentially, the 4 lines. Different people have different calculations because they are plugging it individually. So there may be variances as how steep or how steady your slope essentially slopes are. So it's okay if there's like variation. So the idea is that you want to be able to plot that on some sheet and allow which values across time points and across techniques and temperatures. And hopefully you should be able to get a good result for the generation. For example, 4 different types for the 2 different temperatures and the 2 different. 4.
Yeah, 4 lines.
Yes. Then does that answer your question?
Yeah. Because before I was very, very confused with what exactly was happening in terms of what part of the graph, in terms of separating.
Then that's like it's pretty much like if you follow with the units and follow with the guide that I've provided, you can also see based on the Bible counts as a comes to show here. You can even start off at like 10 to the power 6 or 7 and go all the way to think about 8 or 9. You'll be able to see because like the range you'll notice from what I said on purpose that there's a lowest point of highest, so you try to figure out what the experiments are for the first baseline increments, like 10 to the power 7 to 10 to the power 8, and hypothetically to 10 to the power 9. Any questions? It's a lot of this.
Yes. Also, these are the links operations to operate. Anything on?
You don't have to do, not on the graph.
Sorry, like a thought of
I did, but you don't have to. But I would recommend you have a legend. That like that's okay.
I'm probably not about to propose.
If you can, that'd be amazing. But don't forget both intro and discussion.
And how long are those folks?
3 to 5 sentences for your intro, and for the discussion, go to about 5 to 7.
And if you use Professor Bunt's rubric, is that okay too? Is that really, so how does yours install?
Yeah.
Okay.
Does she show different links for the start of the discussion?
I would say like if you have more expansive points versus very like concise and different, like very specific.
Okay, that's okay. That's okay.
That's okay.
That's okay.
Okay, are we showing our results or as a printout or like a handout or are we?
I want to just also clarify. Actually, you would print it out. Ideally, something on the same line paper like this, and draw out the print. That will make it better for you to actually getting the field and to do the dialogue.
Like taking a picture of that and putting it in my file.
Oh no, you can. This is not for that one. But for the generation time assignment, it actually print this out and hand it in to you with your intro results, calculations, the figures, and the discussion.
Print just, wait, so I print out my duration times. I printed it all out and give this all in physical.
Just like, it's pretty much, you have 2 to 4 pages that you print out. The 2 pages of some paper needed, and then you would have like the intro, results, which is the last year, and then the discussion. To answer,
Can we type out the intro discussion?
I wouldn't recommend it because, you don't want to reset the paper. I mean, believe it or not, I'm.
Wait, do we need references for this?
No. I mean, you can, like, refer to, like.
So can intro on one and discussion on the other?
Yeah, that's
Okay.
That's
I'm worried about this.
No, oh, I understand. Yeah. No problem. But do you have a for both? Why do we put a bottom?
I'm going to replicate the calculation. I just want an example to show you the, let's see if I don't.
That's okay. Let me just see the other case. I just want to make a mistake. One thing I wanted to clarify.
This is that calculation.
Oh, calculation.
If you have to go.
No, this is like one of the parts. That's the part that.
I wouldn't recommend taking photos. Yeah. But
Excel's really good look. You can you can like average them out by like blocks and with of like 2 hours it takes like 10 minutes. That's it.
Thank you so much.
No problem. But if you have any questions, like the sooner you share them, the better.
Okay.
Did I answer your question?
Yeah.
Okay. Any other outstanding ones?
No. Thank you.
All the best. No problem. So I think we did have to go. But let's see. What question?
One more quick question, well, I'm not sure if it's good. Way back, sorry, for the unknown assignment.
Yes.
I got like 3.0, but I really marks. What do I use like the 0.8 marks for?
So I have on Excel with details provided, like a feedback form. Sorry, like the score. Take your pause. Was there anything else? There was one thing that came to mind. I wanted to tell you, but I forgot.
As long as you see everything is okay.
I was just worried there was like 1 or 2 things that came to mind.
Was it data points for one?
I think it was.
I have not been watching the data data point. Usually,
You're doing the means, so that's okay.
Should I rate each point and just underline?
No, that's okay. You don't have to. There was one other thing though. I forgot. Can I just see it whenever you have time? If that's okay. I'm gonna also close this. Your question was about the unknown assignment. Is it okay next week if I show it to you?
Sure, I guess
so. That's okay. The reason being is because I can give it to you, but the problem is the laptop also. So
I know.
like honestly, we have the time to speak. I don't want anyone to feel intimidated, but I also or like
No, I got it
like,
being true to your soul.
and I also want to respect you. Like I understand what you're saying, but I also want to answer your questions here because that way, if you want to see the slides, I can show it to you directly. But nonetheless, let me briefly see. Maybe I'll ask you a question. So viable post-trade entities, just to make sure, I didn't see that it's something here. Having the differences in between those would include
These are the units, so it's still like minutes to negative ones.
if like even your Google Word suffices, I mean technically log on won't physically dimension. So essentially you don't have units attached to you, but if you think about like for the x-axis, the difference is in units, right? So you'll find it comes with units here, and then when you bring to we divide by hope, that's where you.
Okay, so I should include minute like in the denominator.
Yes.
Okay. aside from that, everything is good.
And if you have a range that is like potentially over 15 minutes for your determination, so like 50, 15 minutes, 50, 30 minutes, for your Bible counts, do a similar thing. Try to have that plenty of ranges that are within your timeline.
So do like one for 0 and 16 and then 30 and 60.
That's okay. Okay. I just wanted to make sure you don't want to do it too close. Technically, the French should be consistent, but in case sometimes if you're too narrow, maybe get too high or too low.
A good stretch.
A good stretch.
Okay. Thank you very much.
No problem. Take care. Have a great day. You too. I had so yours was slide your epidermidis. Ethanol, right? No, not. Actually, let me talk about that. I don't know. I just I feel bad the way I will